Hello Interstellar everyone, welcome to class 11 biology. In this video we are going to discuss the remaining portion of meiosis cell division. Right. In our previous videos we have discussed about meiosis 1. We have seen prophase 1 and then we saw metaphase 1, anaphase 1 and telophase 1. And in meiosis 1 we saw that there is a reduction of chromosome number within the cell. In prophase 1 of meiosis 1 we started with the diploid cell. Diploid cell means it contains two sets of chromosomes, one from the father and one from the mother. And by the end of meiosis 1, that is telophase 1, we get two daughter cells with haploid chromosome number. So we get two haploid daughter cells by the end of meiosis 1. So therefore, meiosis 1 is called as reductional division. Meiosis 1 la reductional division la wore. Kachin la wore la wimbana meiosis 1 gi kabla. chromosome gi thanga jomar ti chega takda suyo daughter cell nanglo la so nga dine start cherwa cell di nanglo la chromosome khajo yore la wi ba na 2 n yor it is a diploid cell di nanglo la pala jan le gyo amla jan le gyo cell khaj khare chromosome ni ga yor set ni yor and by the end of meiosis 1 zodu zambala telophase 1 gi thamal le du zambala we get two daughter cell ni ra yor daughter cell ni ga khare haploid la na khonzo gi nucleus nanglo la chromosome ki thanga jomare thajinge ki chromosome ki thanga ga chega takda yor so the chromosome numbers is getting halved. Okay, so that is the end result of meiosis 1. And by the end of meiosis 1 telophase there is a cytokinesis resulting in two daughter cells, and the chromosomes, right, the chromosomes, they haven't extended fully into chromatin. chromosomes 되게 상마 차상마 extend chai 고가 브라그 말에 아니 코 replicate 게 치mar. Therefore the gap between meiosis 1 and meiosis 2 is not called as again interface lag marta khalagre interkinesis lagre seujungso meiosis 1 da meiosis 2 gi parala gap jorwa ila khalagre interkinesis lagre. It is very short and then there is no replication of dna replication yungu mare seujungso so dna gi thanga de pargo marta meiosis 1 da meiosis 2 gi parala seujungso and ta ditsawe jela an khajigre the two daughter cells they will immediately start dividing. meiosis 1 ne azul daughter cell ni ra ro khonsu khajir lamsa divide che go zur kharna divide chidu zambal kharna du gore division dikhare la wemba na meiosis 2 re. And meiosis 2 now again we have got these four stages: prophase, metaphase, anaphase, telophase, and they carry this number from meiosis 2: prophase 2, metaphase 2, anaphase 2 and telophase 2 and here. Okay, so thangbo dita wemba na meiosis 2 gi stage thangbo dikhare prophase 2. Okay, so in prophase 2 of meiosis 2, nuclear membrane it disappears, right? Nuclear membrane it disappears by the end of prophase 2, and the chromosomes which has slightly extended, partially extend chagyo chromosome dige khachire, they will start to become compact again. Okay, chromosome sama khachire rangja compact chadu sobta. So one important thing to notice over here is meiosis 2 it resembles mitosis. So can also mitosis yabu shingi you know meiosis 2 pele lor. So meiosis 2 resembles mitosis, and the resemble chigi obi mena prophase nanglo la khar khar chungu me dige sangma mitosis ki prophase the chibu chigi antela. So what happens in prophase 2, right? The nuclear membrane and the nucleolus it disappears by the end of prophase 2, and the chromosomes they start to become compact again. chromosomes dige sangma khajir yangya compact chayangur and spindle dige sangma form chigi do spindle, right? So ki centrosomes yo cell organelle jangdu sambala centrosomes kan jay ba centrosome double chadur centrosome double chani and khajido they start to move apart and they start extending microtubules. Diche la khare prophase 2 gi jela khare metaphase 2, right? Again in metaphase 2 what happens, the chromosomes they align at the equator la khalor kidik de la warwa cell kidik de la khachire the chromosomes they start aligning over here. Now if you look over here ngazo khane gozu be cell ni ne gozu ba so ngazo cell ni de tyon di nanglo la. So ine chromosome dige sama khajido kimala phangdi do dinalo yo chromosome dige sama khajido kimala phangdi do phangdi junge de surela so nyegi spindle fibers, right? The spindle fibers they push and pull so that all the chromosomes they get aligned at the metaphasic plate. The microtubules from the opposite spindle they get attached to the kinetochores of the sister chromatids. spindle fiber dige sama khwa jar to microtubule dige sama khwa jar sha chromosome gi kinetochores la jar do kinetochores su ga ji kinetochores of the sister chromatids ta dinge de kiran zola are metaphase ta metaphase 1 gi paral difference tissue la so di nalo la ngal pusum gi ngal tang sta le shi ba su ga tang ma sta pusum len tang jo di nalo la kiran zo seu thi gu yo be if you want to clarify the answer over here, right? In metaphase 1 nanglola, there is a pairing of homologous chromosome. Right? There is a pairing of homologous chromosome, and the microtubules from the opposite spindle, they get attached to the homologous chromosomes kinetochores. Sister chromatid lan ke she gumar da khaji gore spindle fibers shomarwe microtubules tige sama khaji gore homologous chromosome gi ani centromere la yo gi kinetochore kan jar gore. So the homologous chromosomes will be pulled apart later on in the metaphase or in anaphase 1. Metaphase nanglo kharchi gorala wimbana the microtubules of the opposite spindle, they get attached to the kinetochores of the sister chromatids. They are chromosome chik madu medu sambal khachi gore microtubule dige sama khachi gore sister chromatid ki kinetochores la jargor chromosome khurang khaji kan chibu marta khaji gore sister chromatids ki kinetochores kan jargor. So diche next khare metaphase 2 gi jela khare anaphase 2. Again what happens, anaphase 2 will resemble that of metaphase ki khare mitosis khaji gi anaphase chi baji gyare anaphase 2 nalo khachi or the centromeres of the chromosomes will split chromosome ni dus nyam do jar de ro kuni ni centromere la jar de ro chromatid ni di centromere la jar de du sambala ta khaji or de la kinetochore la thagwa chik the jar sa ro microtubule chik jar sa ro hijo side de la khajere cystochromatic ki keba de la microtubule chik jar sa ro. So the microtubule, they start pulling. When it starts pulling, the sister chromatids, they will separate khande separate cha wore centromere di break chi wor kuni ni parallel yo centromere jo marwe it splits, and the two sister chromatids, right, the sister chromatids, they separate and move towards opposite pole. So you can clearly see over here anaphase kabla khachi do tade de sister chromatid ni nyam do jar de yo khachi do kuni ni opposite pole la pha du do. So during anaphase, the sister chromatids, they separate and move towards opposite pole. Khajin opposite pole ngo jo du gore la when microtubule gi thengor pha spindle den jo gi pha thengor. So that is anaphase 2. And after anaphase 2 we have got telophase 2. Telophase 2 the chromosomes which were moving towards opposite pole, they have reached their respective poles. End position the nuclear membrane or the nuclear envelope, it starts to reappear. Nuclear envelope starts to form here. Why it happens, how it happens, right? But under microscope you can clearly see that when the chromosomes, they have reached their opposite poles, the nuclear membranes, they start to assemble, right? So the chromosomes get enclosed within the nuclear membrane. And nuclear membrane, the nucleus loga chasa wata lamsa khachigre cytokinesis chungre. So since this is animal cell, there is a formation of cleavage furrow, and there will be formation of two daughter cells, right? Cell ni ne gozu ndu zambala khachigre we get four daughter cells. Cell dine cell ni ragdo, cell dine cell dingi ragdo. So total mazul khazur we get four daughter cells, and these four daughter cells, they are haploid. They are haploid daughter cells. Dine chromosome khazur ne gozu da chik ni sum ne gozu da. Di nanglo lekhare chromosome sum daughter cell rare nala sum. So haploid rangja deidwa. So it has got four. It results in the formation of four haploid daughter cells. The dela important point jorda. Anda badu dela metaphase ki kablarda. Now pay attention over here. Metaphase ki kabla X shaped do diranga chromosome chik lagyo. We call it as one chromosome, which is containing two sister chromatids. Sister chromatid rare langa chromosome lagmewa at metaphase when the two sister chromatids are attached at their centromere. Centromere la jar de zambala ngazu sister chromatid ni dus X shape na yo de langa khal lagyo. It is called as a chromosome. As soon as they get separated during anaphase, anaphase kabla sister chromatid ni de pha tox na chawa darwa lamsa khar cha sare. Now we can call them as chromosomes. Chromosomes of the future cells. Also, sister chromatid khachele ko they got upgraded to chromosomes. Sister chromatid ni looks loga chawada, ta chromatid lagmarda. They are no longer called as chromatid. They can be referred to as chromosomes of future cell. So di induzambala, they la chromosome ching ni sumyo, X shape che chromosome sumyo. They la single strand che yo, chromosome sumyo. Di induzambala, chromosome ki thanga dirangja deyarda. Seu chungswa. So, cytokinesis results in the formation of 4 haploid daughter cells. And all these 4 haploid daughter cells, they are unique. Konzo sama khare loga yer. Konzo li kejo loga yer. Khare kejo loga yer la wimba na. Konzo ki chromosome nanglo la gene composition jo marwe. Rigze ki composition jo rwa. Ti loga yer. Ti nizambar konzo sama unique chadu. They are all unique. Unlike mitosis. Mitosis kabla kharchi all the daughter cells, 2 daughter cells, they are identical to each other as well as they are identical to their mother cell. But here, by the end of meiosis, Meiosis II ki telophase cha vejela, ani daughter cell shira urwa. Daughter cell shiga khare, konzo sangma khare unique re. Unique lade zambala, konzo sangma penju phazu dagma re, sangma loga yer. Khare ki chane dagma re la wimba na, konzo nanglo la gene composition jo marwe. Chromosome ki gene composition di manda chadu. And all these 4 daughter cells, they have the potential to become gametes. Gametes they have all got the potential to become sperms. And if it is inside the female's body, they all have the potential to become eggs. So this is the end result of meiosis II. So with this we have come to the end of meiosis II, telophase II end lelesare. Now let's talk about the significance of meiosis. Right? Let us talk about the significance of meiosis. Sequence Wikipedia you can refer to Wikipedia. Step by step. And I advise you please practice drawing these diagrams. Diagram draw practice chida. Now let's talk about the significance of meiosis. Meiosis la di kachin ke chambure. Mitosis the mitosis ki significance kan ke shemba. Mitosis ki significance kare it is essential for growth and repair. Aimba. Dinangshin de la kare significance of meiosis yore. Significance of meiosis di kare la wimba na. Meiosis ki significance growth and repair na dewa khoro yomarda. Kachin it is a special kind of cell division which happens in sexually reproducing organism, and it happens only with special cells which is involved in sexual reproduction, right? So the significance of meiosis kare, it helps in the conservation of specific chromosome number of each species in sexually reproducing organism. Species specific chromosome thanga jomarwe deep maintain chela. Isi species chi ki yimba na ki nanglo gaji ki cell nanglo la chromosome ki thanga jomarwe specific chromosome number joro di maintain cheya. Mi doji imba na mi ki cell nanglo la chromosome ki thanga jo marwe specific thanga jorwa di maintain cheya. This sui ki chetu gore la imba na sui ki cutting ki chetu gore na because of meiosis. Conservation of species specific chromosome number in sexually reproducing organism. Konjo ki khandes maintain chetu gore la imba na by reducing the chromosome number into half in the gametes. Gametes nanglo la chromosome ki thanga di chega suidu zambala khaji gore ani gamete ni khaji re sperm the egg ni ya jar ndu zambala chega ya domni rangja khaji gore chik charu gore chasan chik charu gore so thanga di khadu ni maintain chedi do. So it is because of meiosis, right? Species they can maintain their specific chromosome number. So that is first significance. And the second significance, which is more important over here, is that meiosis, it increases the genetic variability in the population of organisms. It increases the genetic variability of population organism. Example simple if you have brothers and sisters, look at each other, right? Look at each other. Dagumare, Kejo Dagumare, Dongi Ib Dagumare, what happened is you are all unique, you are all different from each other. The variability the khandeslere through genetic variability through lero. And genetic variability the kajin lere. So stick bar on a palamla chigro. Pu sama chiba ke ora on chena. Loga ki do. Loga ke go jumse khalan chana subu sama nanglola jin manda yor. Jin manda khandes chara la wimba na. Palagi supu nanglola spermatogenesis chungwe kabla gametogenesis spermatogenesis chungwe kabla sperm sangma unique produce chi woro amla gi sobu nanglo la oogenesis chungwe kabla egg sangma manda produce chi woro di indu zambala kira fertilize chungne kira chadu zambala kira unique individual chik char unique sperm ji da unique egg ni ji ya zomni kira unique individual chik char kira jojo ge khare unique sperm ji da unique egg ya zomni khar chare jojo unique individual char aja ge thakara re di indu zambala 이 가진 용거래 meiosis ki cutting 용도 아니 meiosis ki cell daughter cells are unique conditions suido how does meiosis create 4 unique daughter cells from a single cell so conditions suigo la women through recombination process recombination la ji jang ba ngan jogi prophase 1 nang lo la there is a formation of crossing over and crossing over leads to the exchange of genetic material that is called as recombination genetic material phaso exchange chedu zambala kha chore chromosome ni ki parala so genetic material phaso share chedu zambala next chromosome pha mix chagyo jomare this su gi cell na pha drop in cell di khar cha do unique cha do jig manda cha do so recombination is very important with the help of recombination which happens during prophase 1 of meiosis right it helps in the creation of genetic variability within the population of organisms so except in the identical twins himbana genetic variability yomar but sangma na khare genetic variability lead isi organism ji population na ta himbana khi sangma dagmin do mi sangma dagmin do chingdon ro jimen sishin ro jimen hon sangma dagmin do since they are sexually reproducing organisms there is a involvement of meiosis in the formation of gametes right and meiosis it results in the formation of unique gametes which means they have got a unique composition of genes and that will result in the formation of genetic variability in the population and it is a good thing right genetic variability you have got pen nga chimpur organism sangma chikpa chigyan himba ina ro ti yapo yomar so khajin yapo yomar la himba na isi organism sangma chikpa chigyan himba ina evolution yongumar there is no ground or no basis for evolution so variation is important for the process of evolution to happen evolution la de chungwala yarge do yimene yamena ma nyamcha do yimene doye jumare di khandes yungure la through the process of evolution the process of evolution chungwala kharu gu gore variation gu gore example chikte isi thugdan sangma ro mosquito isi thugdan sangma la kheju chipa chigyan yo bimena jin sangma kheju chipa chigyan konju subu gu top sangma chipa chigyan thugdan ro isi nganju gi metor we applied some kind of insecticide tor konju gi population nanglo la genetic variability me sangma individual sangma chipa chigyan yin baina individual chik thug don shi individual sangma shigro that means konju khajeso konju gi species kora mewa chaso inde mayo he chedo there is genetic variability and same goes for us ngazo chedo chipa chigyan re kisi ngazo gi sopu nanglo ga ji immune system na control chengi gi genes so marwe gene sangma mi sangma na chipa chigyan hi mai na neyam chik duks khya ba na khaji re mirig la jo mare mewa cha dur di mayo e chedu we need genetic variability so that evolution favor those individuals who can survive in these tough conditions environmental stress withstand chetung yo ngazo gi gene pool na yo go so these are the two significance of meiosis. Significance is neater ta meiosis ka jigi. Chikhare chromosome thanga maintain cheya species nanglo la. Niwa dikhare species population nanglo la sexually reproducing organism ki population nanglo la genetic variability individual manda chaya jumar niwa nyutop manda yongyo swedi. Di yong imen hachi gore evolution la helps you gore. It helps in the process of evolution. Okay, so with this we have come to the end of chapter number 10, which is cell division and cell cycle. Right. Next time chapter number 13, photosynthesis in higher plants. Okay, so please read about photosynthesis in higher plants. Then you can watch the next video. Okay, thank you.